What the heck? Where did that go? Can somebody please confirm you can hear me?
We can hear you.
Great. Before we begin, are there any questions? Where am I? Not hearing anything. Let me share my screen here. That's odd. I don't have a complete share options for making it available for improved for videos, but hopefully it'll work. All right. What happened there? I'm getting it to share. There it goes. All right. Today is September 30th. And in the lecture, we're supposed to be finishing chapter 1 and beginning chapter 2. We're a little behind schedule, so hopefully we'll get started with chapter 2. I need to change that. The practice quiz has been delayed till Saturday. And you'll learn about that in the lab. Practice quiz 00. Let me change that right now. All right. It's only changed from my version, not the version online, but I'll get that up shortly. All right, I have that we were on the history of microbiology right here, and we talked about Janssen developing compound microscope for Robert Hooke, seeing cells. Did I show you what he saw and why the term cell was used? Anyone?
No, you did not.
Okay, let me explain why Robert Hooke called them cells. When he looked at the cork, thin shavings of cork, he saw, oh, this is not the right one. I want paint. There it is. He saw something that looked like this. And it would be obviously a little more square than I'm showing here. Oh, I'm not doing a very good job making these little looking square, but you're getting the picture. And it reminded him of the rooms in a monastery where the monks had their private room, and this would be like the hallway. And then there'd be a door to each room, which you didn't see a door in the cells of the corkscrew. But the monks called these private rooms, but they called them cells in their monastery. And it reminded Robert Hooke of the cells of the monastery. So he called these shapes in the plant cells a cell. And that's where the term cell comes from. Any question about any of that?
I don't think you were sharing screen.
Oh, yeah, let me know when that happens. I did hit share the screen.
I can see it.
I can see the screen.
I can see it too.
All right, I put share screen. So if you can't see the screen, I'm not sure what the problem is. Let me optimize for video. Now, I didn't get this before. That's why I couldn't optimize for video. That's actually not a video. But hopefully you can see that now. You know, this doesn't look like I'm sharing my screen, am I?
You are. I see it now. Thank
Yeah.
you.
Okay. I'm not sure why my picture is here. Usually when I share my screen, I don't see myself. I see what's on my computer. Anyways, that's why Robert Hooke called them cells. In the history of microbiology, a large part, especially the early history of microbiology, concerned the hypothesis about how life originates. There were two competing hypotheses to explain how life began: spontaneous generation and biogenesis. In spontaneous generation, living things arose by a vital force coming in to non-living matter and then changing it to something alive. And there was the theory of biogenesis that living things only arise from other living things. It was never a question about where a baby came from. Everybody knew that a baby came from two parents. But other life forms, people believe that some of them spontaneously arose. This is in the past. Like over a century ago, people would throw a log into the fire and then a newt would crawl up. People would say, spontaneous generation of the newt. You put the wood in fire and a newt spontaneously generates. And flies emerging from manure, they'd say spontaneous generation of the flies. And the same with maggots arising in a corpse. They'd say spontaneous generation of the maggots. The vital force came into each of these, I don't know, matter, the wood in the fire, the manure in the air, I guess the corpse in the air, and then the maggots or the flies or the newt arose. In reality, the newt was probably in the bark of the wood, and when they put it in the fire and the newt wanted to escape the heat of the flames, and so it crawled out of the wood. We won't talk about what happened with the flies and the maggots, but you can figure it out. Most of these were logical, none of them were scientific, and some were even amusing. Somebody wrote up that if you put sweaty underwear in a jar that had wheat kernels in the jar, that it would spontaneously give rise to mice. In reality, the mice crawled into the jar to get at the wheat kernels, and then they got stuck in the jar because the glass sides of the jar didn't allow the mouse to escape. And the sweaty underwear had nothing to do with the formation of the mice. A brief sidetrack, they thought that microorganisms spontaneously generated because whenever they made a tube or a container of nutrients, after a while, it would change from this type of a picture. Let me blow that up. Where the media was clear, and then after time it would become cloudy, and they'd see microbes causing this cloudiness. And that we call microbial growth in the tube. And this always was observed, and they thought that the microbes were spontaneously generating in the tube of media. In reality, the tube was not sterile, the air was not sterile, and so microbes fell into the tube of media, as well as the microbes in the tube, and then over time they grew, causing the turbidity. and that we call growth. This is clear, no growth. The first evidence against spontaneous generation happened by the experiment by Francesco Redi. And this guy you should know. You don't need to know when he did this, but he also made the first controlled scientific experiment and then wrote it up. He had three different conditions. One condition where he had a jar completely sealed, and he had put meat in the jar and then sealed it. And he found no maggots in the meat. And the people who believed in spontaneous generation said, you sealed the jar. The vital force could not get into the jar and give rise to maggots. And then he had also an open jar, which had the maggots appear. But then he had a jar that he covered with a fine wire net, and he found no maggots in the jar. And the fine wire net or meshing allowed the air to get into the jar, but no maggots occurred in the meat. And this was the first evidence that the maggots did not arise by spontaneous generation. Any question about that? It's also the first controlled scientific experiment that was ever written up. After this time, everyone agreed that complex living life forms did not spontaneously generate, that they had to come from the theory of biogenesis, arise from another other organism, meaning the maggots were a complex life form, multicellular, and that they came from actually flies, which landed on the meat and then laid their eggs on the meat, which gave rise to the maggots. However, microbial growth occurred in the flask, and so people argued that microbes could spontaneously generate. And this went on for, what was that, 1668? You don't need to know the years. Till 1861, almost, geez, that's almost 200 years. Is that right? Yeah, almost 200 years after Francesco Redi. Finally, Louis Pasteur proved that microbes did not spontaneously generate. And before I talk about Louis Pasteur's experiment, I need to talk about the beginning of microbiology. The first microbes were observed by Antony van Leeuwenhoek, shown here, who was actually an amateur biologist. He was actually a person who, what do you call that, a tailor, made cloth and all that. And he made microscopes to study his cloth. And he's the first one who looked at things and discovered microbes. He actually looked at microbes from scrapings from his teeth. He found microbes in rainwater. He found microbes in peppercorn infusions. And he found microbes in the water obtained from the River Thames, which was the water that supplied the city of London. So the people in London weren't happy to hear that they were drinking a bunch of microbes. The reason why he saw microbes and other people did not was because he was the best lens grinder of his time, and he made the best microscopes of his time. This is actually a microscope that he made. It's a simple microscope, only having one lens to magnify the objects. And because he only used one lens and he was the best lens grinder, he was able to see things that other people were not able to see, including the microbes. He was even able to see cells so small they were not seen for almost a century later. And that's because his microscopes were so good, you could see these small cells in clarity, and the other microscopes of the time made by Janssen, which were compound microscopes, were not built good enough to be clear enough to see these cells. You can look here. He drew a picture of bacteria cells. There's a rod, and there's its movement. There's a spirilla. There in figure E, we have two cocci. And then in figure F, rods, with that one being a bent rod or two rods stuck together. So these are the first bacteria that were ever seen, and they weren't seen again for nearly a century later. After a century, the people started improving the microscopes. They got better with time. And after a century, the focusing of the microscope got good enough that you could actually see the bacteria. Any question about any of that? All right, I may be butchering his name. I'm not Dutch. He was a Dutch. And Tony van Leeuwenhoek is the first microbiologist. The beginning of microbiology now began. All right, you don't need to know this, but in 1858, Rudolf Virchow proposed the cell theory. It's actually an extension of the cell theory. He actually wrote it up, that all living things arise from only pre-existing living cells. This argument about whether microbes could spontaneously generate continued until 1861, when Louis Pasteur showed that microbes did not spontaneously generate, meaning he disproved that hypothesis and therefore confirmed the theory of biogenesis. What Louis Pasteur showed was that microorganisms are present in the air that can fall out of the air and then contaminate a tube. And you could also kill microbes by destroying them by heat. So Louis Pasteur showed that. And what he did was his famous, ingenious swan neck experiments, where he got a container with a long neck. This is called a flask. And then he poured media in it, and then he heated the neck of the flask in a swan neck like that, and then sterilized the media inside by boiling it. And then he allowed it to cool, and he found that this flask never became contaminated with microbes unless he put media all the way down here and then brought the media back up. What happened was microbes were falling into the open part of the tube right here, so the vital force could get in and create the spontaneously generated life. But the vital force obviously did not. And the microbes fell in, but they were trapped right here by gravity, and they did not go up the tube and then contaminate the media, unless you put the media down into this portion here and then bring the media back. Any question about Pasteur's experiment disproving the theory of spontaneous generation? All right. Beginning with Pasteur's work, a bunch of other discoveries occurred in microbiology, including the relationship between microbes and disease. This became known as the germ theory of disease, that microbes cause disease. This also gave rise to the theory of immunity, that our immune system could protect our body when microbes invaded a body. The immune system could then attack it, giving rise to immunity and then protecting the body from those microbes. This also gave rise to the theory of antimicrobial drugs, that there are drugs that you can use against microbes to cure a patient of an a microbial infection, for example. Any question about any of this? Shortly after Louis Pasteur's work, Louis Pasteur developed the basic idea of a aseptic technique. You need a aseptic technique in microbiology, otherwise you will be getting contamination of a pure culture, and then you won't be studying one species, you will be studying multiple species. I've already mentioned before, if you have multiple species in a tube and the tube turns yellow, you don't know which species caused the tube to turn yellow. And so you cannot say, for example, like Staphylococcus epidermidis, one of the species in the tube, caused the tube to turn yellow. So you need aseptic technique so you can keep a culture pure. And then Louis Pasteur came up with a very complex way of developing a pure culture, and that is he did limiting dilution of a liquid culture. See if I can find paint again. So he made a serial dilution. I'm only going to do a few of these, but he would do like 5 or 6 of them. And the first would be diluted 1 to 10. The next dilution, 1 to 100. I'm having a hard time writing this. So let me get that one there. So the next one would be 1 to 100. And the next one, 1 to 1,000. And then you would take an aliquot of each of these dilutions, put it in, well, no, actually, he would then take an aliquot of each of these dilutions and put it into 10 different tubes. So 10 different tubes of 1 to 10, 10 different tubes of 1 to 100, 10 different tubes of 1 to 1,000. And if he got only one of the 10 tubes with growth in it and 9 of the tubes with no growth in it, he correctly concluded that the tube with growth in it, and only one of them, meaning 9 with no growth in it, but the tube with growth in it was likely to be a pure culture, meaning one formed by one cell, or at least a few cells. So it was a pure culture. And then the other dilutions that had more than 1 out of the 10 tubes with growth in it were likely to be contaminated by a non-pure culture. Any question about any of this? All right, that was a complex way of getting a pure culture, but it was the first way microbiologists had. About this time, Dr. Koch developed a much easier way of obtaining a pure culture, and that is he streaked cells, mixed culture of cells, onto a solid surface, getting isolated colonies. And if you streak the cells onto 2 separate plates, meaning you get an isolated colony on one plate, you take from that one isolated colony, streak it for isolated colonies on a second plate, the colonies on the second plate are likely to be a pure culture. Now, admittedly, it's not 100% guaranteed that all of the colonies on the plate will be a pure culture, but it's greater than 95% chance that they will be a pure culture. And that's good enough for microbiological use. You never get 100% chance that you have a pure culture, no matter how many times you streak it out. There's always a chance that one colony you streaked from was formed by 2 or more cells, and then you streak it on another plate, and the colony you pick from that plate was also formed by 2 or more cells, etc., no matter how many times you do it. So you never get 100%, but each time you get closer to 100%. Anyways, that was a very easy way of getting a pure culture. You just streak it out on solid media, First time, get an isolated culture, take that isolated culture, put it on a second plate, and then get isolated colonies, pick from one isolated colony, and then you likely have a pure culture from that one isolated colony. Around this time, somebody in Dr. Koch's lab actually developed nutrient agar, a very good, solid growth medium or liquid growth medium, doesn't matter which, for growing microbial growth. And we've talked about nutrient agar being a general purpose media, growing many different organisms. As long as the organism is not fastidious, meaning one that requires specific additives to it, it likely will grow on nutrient agar or any general purpose media. And then around this time is when Sergei Winogradsky developed the idea that microorganisms evolve in the biogeochemical cycles, such as the nitrogen cycle we've already talked about. He also discovered the sulfur cycle. And the reason why he was able to find these cycles is because he was not studying pure species. He was studying microbes growing in a community of many different species growing together. You need the many different species to get the nitrogen cycle to occur. All right, Louis Pasteur also went on to show that microbes were responsible for fermentation. He showed that alcoholic fermentation, the conversion of sugar to alcohol, happens in the making of beer and wine, and that's what gives beer and wine its kick. He also showed that microbial growth could spoil the beer and wine or other food. In the bacteria, other microbes could grow in the alcohol, like the beer and the wine, and then spoil it by changing the alcohol to vinegar or acetic acid. The concept that spoilage bacteria could be killed by heat, it was not hot enough to evaporate the alcohol in the wine, gave rise to pasteurization. Louis Pasteur actually developed pasteurization for the beer industry because they asked him, why did our beer spoil, becoming sour and undrinkable? And can you come up with a way to prevent that from happening? And he came with pasteurization, the application of a high heat for a short period of time that did evaporate a little bit of the alcohol, but not all of the alcohol. And pasteurization is used in a wide variety of foods today, most commonly associated with milk, but it is used in many other food products today. Any question about any of that? I thought I talked about pasteurization. I guess later I talked about pasteurization. All right, fermentation, the realization that microbes can affect an organic matter and change it to a different organic matter, meaning change sugar in whatever grain you're growing to make the beer or the grape juice to make the wine, led to the question, could microbes have similar relationships with plants and animals, and might microbes cause disease? This idea became known as the germ theory of disease. Not one person proposed the germ theory of disease. It developed over time by many different people. Originally, it's a very important theory about how disease could occur, because originally people believed that poisonous vapors were breathed in and were responsible for causing an infectious disease. Like cholera, people believe people walking through a swamp breathed in the poisonous vapors of the swamp and then got cholera. In reality, the cholera was present in the swamp, was in the air, and then people breathed in the cholera microbes causing the cholera. Any question about any of that? Two people who are important in the germ theory of disease. Ignaz Semmelweis, he advocated hand washing to prevent the transmission of disease by pregnant women during pregnancy. Usually the women, or oftentimes the woman, came down with either postpartum fever or puerperal fever. And he showed that if you had hand washing, meaning the surgeon and the nurses washed their hands, this greatly reduced the transmission of disease to the pregnant mother. However, the either the vice president or the president, sorry, I don't remember who that was anymore, probably the president of the hospital, was so insulted that Schimmelweiss was suggesting that doctors wash their hands, that he actually fired Schimmelweiss from the hospital, even though he had evidence to show that when you hand wash, the pregnant woman was much less likely to get an infection as well as die an infection. And another person who was important in the germ theory of disease was Joseph Lister. He used chemical disinfectants to prevent infections during surgery. He dramatically reduced surgery-related infections, proving that surgical infections are caused by microbes. He actually used benzene or carboxylic acid to treat the patient's skin, as well as maybe the surgical instruments, to clean them, not exactly sterilize them, but make them very, very clean, reducing surgery-related infections. Any question about any of that? And benzene was just the best agent he found. We have much better agents now that we can use in surgery. The germ theory of disease was actually approved by Robert Koch. So Robert Koch did not propose the germ theory of disease. He essentially proved it. And we never prove anything in science, but he showed that the bacterium Bacillus anthracis causes the disease anthrax. And then he wrote up the experimental steps required to prove that a specific microbe causes a specific disease, and this became known as Koch's postulates, the steps we use to show that a given microorganism, meaning of one species, causes a given disease. And by this, he provided evidence that the germ theory of disease was correct, at least for infectious diseases. Any question about any of this? This is the first time we'll talk about Koch's postulate in this class. We will talk about this subject 3 times during the class, meaning it's so important we'll talk about it at 3 different times. So you can expect a question about Koch's postulate, not only on the quiz, maybe for chapter 1, but you can expect a question on the final on Koch's postulate because it's such an important, tool in microbiology. Around this time, Edward Jenner developed the first vaccine. He made the first vaccine against the disease smallpox. He inoculated a person with the cowpox virus and then discovered that that person was protected against later smallpox. The reason for how cowpox protected an individual against smallpox was not understood at the time. And so nobody was able to make another vaccine for about a century, maybe more than a century later. And Edward Jenner had no evidence that cowpox prevented smallpox, but he did experiment on children. He injected 2 boys, one being his own son, with the cowpox virus. And most people would say that would be unethical. You shouldn't inject something into a person, especially a child, but especially a person, to prevent disease. And it's true, it did work, but Edward Jenner had nothing to go with other than his own thinking, his own reasoning. He was told about cowpox by actually a milkmaiden, and I guess smallpox was happening in the village or a city Edward Jenner was in, and a milkmaiden came to him and said, "Oh, I don't need to get smallpox. I've had cowpox because I'm a milkmaiden," and they frequently got cowpox. And you can read about the poetry back in these days, and people would write poems about the beauty of a milkmaid, and that's because they never got smallpox, which made um, uh, indentations or um, scars on their face from the smallpox disease. Pasteur was actually the person who experimented with vaccines and actually showed that the reason why cowpox was protecting against smallpox is because cowpox was genetically related to smallpox. And when the immune system made a response against the cowpox virus, some of that response also reacted against smallpox. So the two viruses were related, and that's why the reaction against cowpox protected against smallpox. With that, people understood how vaccines worked and how to make vaccines, and other people started making vaccines, including Louis Pasteur, which he made actually a number of vaccines. The only one I know off the top of my head is he made the first rabies vaccine. The term vaccination is actually derived from the word vacca, a word meaning cow, from the cowpox virus being a vaccine against smallpox. And this protection against the vaccine we call immunity. Any question about any of that? All right. Chemotherapy is the treatment of disease with a chemical. Nowadays, chemotherapy is used almost exclusively for treating cancer, but strictly speaking, it is the treatment of any disease with a chemical. We will use the term chemotherapeutic agent to treat an infectious disease, or synthetic drug, or an antibiotic to treat a microbial caused disease. An antibiotic, strictly speaking, meaning by the textbook definition, is a chemical produced by a living organism that inhibits or kills other microbes. And this is a textbook definition, not the way that the term is commonly used by the public, or even the way it's commonly used by clinicians. For example, penicillin G is an antibiotic. It is made by the Penicillium mold, and so it is an antibiotic. But carboxylin is totally man-made in the lab, so it is not an antibiotic. It is an antibacterial drug, according to the textbook definition. And then even ampicillin, which is made by an organism but then chemically modified by humans in a lab, is not an antibiotic by the textbook definition. This is not the way I will use the term antibiotics, because it's not the way most clinicians use the term antibiotic, meaning I'll use it as it's commonly used by the public, meaning carboxylin is an antibiotic, at least the way I'm using it. Any questions about any of that? Paul Ehrlich found the first drug, chemical, to treat a disease, syphilis, in 1910. he called this a magic bullet, a chemical that could treat a infectious disease and then cure the patient of the infectious disease. The magic bullet he found was a synthetic arsenic drug called salvarsan. And he found that in 1910. Curiously, in his lab, people put up above his lab a sign saying, "Abandon all hope, ye who enter here." And that's because he was just going through one chemical after another, trying to find one that was an antibacterial drug. And salvarsan was something like the 500th chemical that his lab tested, meaning he tested about 500 earlier drugs, and they were not antibacterial drugs. Any question about any of that? So Paul Ehrlich's the first person to find a chemical that could be used to treat a drug. However, that was not the first antimicrobial drug ever discovered. Quinine from the bark of the cinchona tree was the first drug that was ever found, and it was used to treat malaria. Does anybody know when the bark of the cinchona tree was first used in Europe to treat malaria? Anyone have a guess? Come on, just guess.
It's 500 BC.
That's a little too early, but at least you're on the right point there. No, the bark of the cinchona tree was never discovered until Europeans found it in the native found it in the jungles of Peru, and so obviously it was after Columbus discovered America. It was actually quinine in the bark of the cinchona tree was first used to treat malaria in the 1600s, I believe 1660 or 1670, something like that. And that's just because the Jesuit missionaries going into Peru discovered that the natives took the bark of the cinchona tree to treat their malaria, and they discovered that it worked, and then they took the bark of the cinchona tree back to Europe. Who knows when the natives of Peru first discovered the bark of the cinchona tree being able to treat malaria? Any question about any of that? All right. So Ehrlich found the first drug to cure a bacterial disease, curing syphilis, and that happened in 1910. You don't need to know the year. You just should know that Ehrlich found the first drug to cure a disease. Alexander Fleming found the first antibiotic where he streaked out his plate, and he was a sloppy microbiologist, and mold spore got on the plate, and then he left the plate at room temperature on the counter. And that was important so that both the bacteria and the mold could grow. If he put it in the incubator, only one of them could grow. I don't remember which one, but only one of them could grow. And he then came back and found his plate contaminated, and most people would have thrown it away. But Alexander Fleming looked at it carefully, and he saw that there was no bacterial growth in a ring around the colony of mold. And he correctly concluded that something was coming out from the mold colony, preventing the bacteria from growing. And he called this penicillin, after the penicillium mold, which was the species that, or genus that caused the colony of mold on the plate. And today we call it penicillin G, the first antibiotic that was ever discovered. Alexander Fleming was not able to chemically purify penicillin G, so it was not the first antibiotic that became clinically available to treat a drug. You couldn't give penicillin G solution to patients because it had obviously mold spores in it and then all the media in it. And that would be fairly toxic to consume it, as well as the penicillin G was not that high of a concentration. So the first clinically available antibiotic was sulfonamide, one of the sulfa drugs. It was a man-made drug synthesized so they could purify it and became the first clinically available drug. You don't need to know when, but in 1935. It wasn't until the early 1940s that people were able to purify penicillin and then grow it up, purify it, and then test it by injecting it into a patient. If there's time, you can ask me about the story of the first two patients treated with penicillin. I don't have time today, but if ever we have time, you can remind me to tell you the story about the first two patients treated with penicillin G, actually. All right, let's change gear a little bit and talk about microbes and human disease. Microbes were once classified as plants. If you remember, Carolus Linnaeus classified all living things, including bacteria, as a plant or an animal, and bacteria were classified as plants. Hence the term flora for microbes. If you ever hear me use the term flora or microflora, that was an older term that I was taught when I took microbiology. It has now been replaced by the term microbiota. And you can correct me if ever I use the term flora or microflora, because I should be using the term microbiota. The normal microbiota are the microbes that normally present in and on a animal, person, or plant. And the normal microbiota are a community of microbes found on all animals and plants. They form a symbiotic relationship with the host. And there's many different species growing in that microbial community. The normal microbiota, like the human normal microbiota, is central to our own welfare. Any questions about any of that? We do have a different normal microbiota between different species, like there's a human normal microbiota and a cat normal microbiota. For example, on cats, Bartonella henselae is a part of the cat's normal microbiota. If Bartonella henselae is on a human or in a human, it causes disease. And we call that cat scratch fever, the disease. Any question about any of that? The normal microbiota is usually found on or in the skin, the oral nasal cavity, the respiratory tract, the digestive tract, and the urethra, at least the lower urethra. The microbiota is not normally found in the blood, internal organs, or internal tissues. The normal microbiota are important for humans, like the normal microbiota of humans, are important for humans that the normal microbiota can prevent the growth of pathogens. The normal microbiota can compete for resources, preventing the pathogen from growing. It can also take up the space, and if there's no space for the pathogen to colonize, it can't get established on the host. The normal microbiota can produce nutrients and growth factors like in humans, a significant portion of the folic acid and the vitamin K that we get is made by the normal microbiota in our gut. The normal microbiota are important for digestion. I think everybody knows that E. coli helps us digest our food. And then the normal microbiota in the stomach of a cow can allow the cow to digest the grass. Actually, the cow itself cannot digest the grass. It needs its normal microbiota to digest the grass. The normal microbiota can also secrete toxins that inhibit the growth of pathogens. And we'll talk a little bit more about that later in the term. Any question about any of that? So the normal microbiota do not normally cause disease, at least in a healthy human being. However, it can cause disease in certain circumstances, such as an AIDS patient who lacks an immune system. The normal microbiota can cause disease. It can cause disease too if the normal microbiota is growing in areas of the body it should not grow, meaning E. coli should be in our intestines. If it's growing elsewhere in the body, like in the ear or in the lungs, it is likely to cause disease. Any question about any of that? All right. As I've stated, infectious diseases results when a pathogen overcomes the host resistance and then grows in the host, causing disease. And infectious diseases are caused by pathogenic microbes. A pathogen is something that can cause disease. Any question about any of that? All right. An emerging infectious disease is a new or changing disease that is either increasing or has the potential to increase in incidence. Can anyone name an emerging infectious disease? What's a new and emerging infectious disease?
How about bird flu?
Bird flu is a disease that they're watching because it has the potential to increase in incidence. So it is an emerging infectious disease, and they're watching that carefully because if bird flu ever spreads from person to person, it would be a seriously dangerous disease in humans, meaning bird flu has a high incidence of killing humans, and fortunately, it cannot spread from person to person because among birds, it actually follows the fecal to oral route, meaning the bird has to eat it and then become sick and then sheds the virus from the feces. And that's how the bird picks it up. And in humans, it currently does the same. And that's why bird flu does not easily spread from person to person. If ever it were to change and become aerosol, meaning spread by the air, it would be a serious new human emerging infectious disease. Another emerging infectious disease which just emerged was COVID-19. It's still considered new, and so that's why it would be an emerging infectious disease. All right, that's it for emerging infectious disease. I've got some slides discussing it, but I'm only going to ask you to know the definition of emerging infectious disease. Any question about chapter 1? If not, let's start chapter 2. Boy, my computer is slow. Come on. There we go. All right, chapter 2, showing you the major goals and a rough outline. Know the terms. What's an organic compound and an inorganic compound? What is a hydrocarbon? What is a polar and a non-polar molecule? What are hydrogen bonds? What's an acid, a base, and what is the pH? Also in chapter 2, be able to explain with respect to the 4 major biological molecules what the relationship is between a monomer and a polymer and what the characteristics of the monomers and the polymers are, meaning what are the characteristics of the 4 major biological molecules. And then describe the composition, structure, and function of the 4 major biological molecules, and then give examples of each of the 4 major biological molecules. Can anyone tell me what 3 of the 4 major biological molecules are? And 3 of these major biological molecules you can find on any cereal box, on almost any item of food.
Carbohydrates, lipids, proteins.
Yes, those are the 3 major biological molecules you can find on a cereal box. The 4th major biological molecule is nucleic acids. Any question about any of this?
Yeah, I have a question.
Uh-huh.
What portions of chapter 2 are we supposed to read in the textbook, or just the
The whole chapter.
whole chapter?
Yeah.
Whole chapter.
Except for chapter 10, I have the whole chapters. So I expect you to read the whole chapter for everything except for chapter 10. Is that true? I think that's true for everything, yes. All right. Go back to the scale of things. So in chapter 2, you should know the scale of things. You should know that humans is one of the largest thing we'll study. Obviously, elephants and whales are bigger. Then a chicken egg, that's actually the largest cell you've probably ever seen, the yolk of the chicken egg. Frog eggs, that's about the smallest cell you can see with the human eye. It is possible to get a little bit smaller than that. Eukaryotic cells, next smallest thing. Bacteria and prokaryotic cells, the next smallest thing. Viruses, smaller than a prokaryotic cell. Smaller than a viruses, we have the large biological molecules like large proteins, large lipids, large lipids or fats. And then we have small molecules. They're smaller than a large molecule. Water would be an example of a small molecule. And then smaller than a small molecule, we have atoms. There are things smaller than an atom, but we're not going to talk about them in this class. Any question about any of that? You do not need to know the exact sizes of things, but you should know the relative rank of the things. You should also know a little bit about what you can see with the light microscope and what you cannot, like viruses are too small to be seen with a light microscope. Any question about any of that? There is a mistake in this graph here, and I don't know why Pearson never corrected this, because he's got 1 micrometer in here twice. Once there and once there. That shouldn't be a micrometer right there. That should be a millimeter. All right. What are organic compounds? They're typically complex molecules structurally. They typically have carbon in them binding the molecule together. It's usually the molecule is most of the time held together by a carbon-carbon bond. Carbon is important because it can bind to 4 other atoms. And then the bonds of an organic compound are mostly chemically covalent bonds, holding the molecule together. An inorganic compound typically lacks carbon. That's not always true, but oftentimes it lacks carbon. Inorganic compounds are required by living organisms for the organism's growth, repair, maintenance, and reproduction. An example of an inorganic compound are oxygen, water, and carbon dioxide. These are all inorganic compounds. They are simpler than a organic compound, except for carbon dioxide. They don't have carbon in them. Obviously, like I said, carbon can be in the inorganic compound, like carbon dioxide is an example. And the carbon actually holds the oxygens together too. And then water is one of the most important and abundant molecules in all living life forms. So water, the most important and abundant molecule in you. Any question about any of that? Organic compounds always contain carbon and hydrogen. Many organic compounds contain oxygen, and they may contain other elements as well. All right. Any question about organic compounds versus inorganic compounds? If not, let's talk about inorganic compounds. They're simpler than an organic compound. We're going to talk about water. Water is a polar molecule. I'm going to call water having partial charges, meaning the hydrogen has a partial positive charge, the oxygen has a partial negative charge. Tutori doesn't use that term. He calls it a slight charge on positive charge on the hydrogen, a slight negative charge on the oxygen. The polarity of water happens because oxygen pulls the electrons of the chemical bond, meaning each chemical bond is formed by 2 electrons. Oxygen pulls on those electrons stronger than hydrogen, so the electrons spend most of their time, or at least majority of their time, around oxygen, giving oxygen a more negative charge than hydrogen, which has a partial positive charge. This is not a complete charge where hydrogen has a +1 charge and oxygen a -2 charge, for example. It's a partial charge. Any question about that? All right, so the polarity of the water molecule allows different water molecules to bind together. And that is the partial negative oxygen will be attracted to the partial positive hydrogen of another water molecule. And that attraction we call a hydrogen bond. That hydrogen bond is very important. Can you guys see this liquid water here? How the water changes shape to fit the container? I'm giving it, can you see that? I don't know if you can see it. Anyone?
Yeah, we can see
Yeah.
it.
Okay. The reason why water is sticking together and then changing its shape is because of the hydrogen bonds holding the water molecule together. The reason why the water is sticking together, meaning it's not flying out as gaseous water, is because of the hydrogen bond holding the water molecule together. The reason why it's a liquid and can change its shape is because this hydrogen bond is weak and can break and then reform or allow another water molecule to form a hydrogen bond to write this hydrogen right here. And that allows the water to be a liquid and take different shapes of the container. If we were to stabilize that hydrogen bond and it would never break, we would have solid water, and that we call ice, where the A hydrogen bond never breaks because the individual water molecules don't have enough energy in them to break that hydrogen bond. And that we call ice, where the water becomes a solid. And as you know, a solid cannot change its shape to fit the container. Anyways, that's water, a polar molecule. You should note that water has two chemical bonds. The hydrogen bond between different water molecules, this water molecule and this water molecule, and then the covalent bond between the hydrogen and the oxygen of a single water molecule. Any question about any of that? I think I have a little video here, but it's not very good. Let's see if it plays. Oh, I have to shut this down. Not getting it to play. There's a water molecule. A bunch of water molecules. Hydrogen gas. Oxygen. This is showing you the formation of water, which I'm not showing you here, but and there's a water molecule. It has a partial positive charge on each hydrogen and a partial negative charge on the oxygen. And that can form water, forming a hydrogen bond between the partial negative charge and the partial positive, and that's called the hydrogen bond, and it's breaking and reforming all the time. And that's why the water is a liquid. If that hydrogen bond were to break and never reform, we would then have gaseous water. And that's what happens when you boil water and then evaporates. The water molecule changes from liquid state to a gaseous state. Any question about any of that? Water is a good solvent. It's a good solvent because it can dissolve any other polar molecule as well as ions. So something that is ionic can dissolve in water, and that's why water is a good solvent. In the case of sodium chloride, which is an ionic crystal held together with a negative chloride ion, and that's a -1, and a positive sodium ion, +1. It's held together by ionic attraction, meaning the positive sodium is attracted to the chloride, and then it forms a crystal so that, here's a good example, this positive sodium will crystallize, form a crystal with each, be attracted to each chloride ion here because of the negative attraction. And that makes salt form a crystal. And if you ever had a big chunk of salt, that would be an ionic crystal held together by ionic attraction. Obviously, the little grain of salt, the grain itself is held together by ionic attraction. And you can put the grain of salt in water, and then the water molecule will orient so that for the positive sodium ion, only the oxygen will be facing the positive sodium ion to put the sodium ion in solution. So this sodium ion, the water molecules can come around it. The oxygen will be attracted to the positive 1 sodium ion, and then the water will pull out the sodium ion, bring it in solution, and a whole bunch of water molecules will have the oxygen facing the sodium, and that puts the sodium in solution. And then the chloride ion with a negative 1 will have the same. The water molecule will come up to it, and the hydrogens will orient to the chloride ion, and then it'll surround the chloride ion and then pull it out of solution. More water molecules will come around it with the hydrogen atoms attracted to the partial hydrogen plus atoms attracted to the negative 1 chloride ion, and that will bring the chloride ion in solution. And that's why the crystal of salt will dissolve in the water. Polar substances do the same. It's just that they will be oriented, the negative water will be towards the partial positive of the molecule. Like the reason why sugar dissolves in water is because the negative of the water will come up to the partial positive charge of the sugar, and then the positive hydrogen will come up to the negative charge of the sugar, pulling the sugar molecule in solution. Any question about any of that? So polar water substances dissociate in water, forming solutes, and that we call a salt crystal. This we call salt water, a solution containing both water and sodium ions and chloride ions. Any question about any of that? All right. Water is a good solvent because so many things can dissolve in it. Polar molecules are hydrophilic. That literally means water, hydro, and then philic, lover. But what we mean by hydrophilic, not water lover, but something that likes being around water and will dissolve in water. All polar molecules and ionic molecules are hydrophilic. A nonpolar molecule will be repelled by water, and that we call hydrophobic. Literally, this means water and fear of. So fear of water. Obviously, the molecule does not fear the water, but it does not dissolve in it and will be repelled by the water molecule. An example of a nonpolar molecule, which is also called the hydrophobic molecule, would be oil when you put it in water. As you know, when you make salad dressing, you put your olive oil and then your seasoning and your vinegar, and usually put in some water, and then you mix it all together. The seasoning and the water and the vinegar will all mix in with the water, but the oil will separate out and will be repelled by the water. Any question about any of that? And that's because oil is a non-polar molecule. It is hydrophobic. In the show here to make sure I said everything there. Another example would be if you put butter fat in water, the fat would be repelled by the water. Hydrogen bonding in water makes water excellent at buffering the temperature. I think you guys all know this because you're either in Western Washington or Western Oregon, and our temperature is buffered by the Pacific Ocean, the water in the Pacific Ocean, which also gets in the air and helps buffer the temperature. The reason why water is good at buffering the temperature is because water absorbs heat in two ways. It takes energy to break the hydrogen bonds between the water molecules, and then the water molecule itself can actually absorb the energy. And that's why in the western part of Oregon and Washington, our temperature is buffered. In the eastern part of the states, the temperature is not buffered, and the summers are much hotter, and the winters are much colder. And it's because water is not buffering the temperature in eastern Oregon, at least as much as in western Oregon and western Washington. Other molecules, like oil, can only absorb energy in one form, and that is the molecule itself can absorb the energy and then vibrate. And the more the molecule vibrates, the more the molecule absorbs the energy, the more the molecule moves. That we actually call the temperature of the oil. So if you heat the oil, the molecules are going to move and vibrate much more faster, and that's why the temperature will rise. That happens to water as well. When we heat water, some of that energy is absorbed by the molecules, which causes the molecules to move and vibrate faster, and that's why the temperature will rise. But some of that energy will be taken to break the hydrogen bonds, and the hydrogen bond will absorb that energy instead of the water molecule itself. And that is why if you give 100 joules of energy to 1 cup of water and compare it to 100 joules of energy to 1 cup of oil, the temperature of the oil will raise to a higher temperature from the 100 joules than the cup of water by the 100 joules. And that's because some of that energy will be used to break the hydrogen bonds, and that will cause the water molecules to evaporate. But the water molecules which are evaporating from the water are actually the hottest water molecules. So when you put energy into water molecules, and then the water molecule has a whole bunch of energy, that will be the water molecule that will evaporate. And that means water is actually very good, not only at buffering to the temperature, but also in cooling, keeping cool the water or a person. When we sweat or perspire, it's going to be the hottest water molecules which will be leaving the person and evaporating, taking the heat away with the water molecule that's evaporating. Any question about that? So there are two things that absorb energy when you apply energy to water. Most other things only can absorb water or energy by one thing, meaning the vibration of the molecule. Any question about that? So oil only has one thing really absorbing the energy. Cement only one thing. Metal only one thing. Most other things only have one thing absorbing the energy, but water has two things absorbing the energy. All right, any question about anything? If not, I'm going to break it here, and we'll talk about water splitting up in the next lesson. I'll see you at 6:30 for those in the 6:30 lab. Any questions before I go? All right, I'll see you.